and then they go through the dark reactions to create sugar. What do we create at the light reactions? What do plants create during the light reactions? Yeah, they create ATP, right? They create ATP and NADPH. And then they create sugar to create ATP. Doesn't that seem foolish? I mean, they have a whole other series of reactions to create a molecule that ultimately will be converted into something that they make in the light reactions. Doesn't that seem bizarre? Why would they do that? And the answer is ATP is incredibly unstable, and you can't store it. Once you make ATP, you have to use it. And so plants are able to take ATP and convert it into a molecular form of energy so that they can store it for a long period of time, so that when they need it, they're able to metabolize that sugar to make more ATP. It's terribly inefficient, but it doesn't matter because solar energy that they're using to drive this process, it's free. They don't have to expend any energy to harvest that light energy. Okay, so let's think about cellular respiration in its entirety. How many ATP do we make in all of cellular respiration?
18.
18? It's close to 18. How many did we make just at chemiosmosis? 34 to 36, right? And how many do we make in electron transport chain? 0. How many do we make at Krebs cycle?
2.
2, right? A total of 2. So that's 36 to 38, right? And then how many do we make in glycolysis? 2. So that's 38 to 40. But is that how many we make? Or is that how many we gain?
That's how many we gain.
That's how many we gain. How many do we actually make?
40.
40 to 42. With a net of 38 to 40. Cool? Okay, so from a single glucose molecule, we can make like 42 ATP if things are working optimally. Now the question is, why is there this range? And the answer is because we make 2 different kinds of electron carriers here. We make NADH and FADH2. And electron carriers, while they do the same thing, do not produce the same amount of ATP. And the reason is, when NADH drops its electrons off, it drops it off at the top of the electron transport chain, the very top protein. When FADH2 drops its electrons off, it drops it off down here at the second protein. So FADH2 drops off its electrons, becomes FAD+, and because it's dropping it off farther down the electron transport chain, it doesn't transport as many protons, and it doesn't drive as much ATP synthesis. Right? But that doesn't really explain why there's a range. All we've shown is that there is a difference between NADH and FADH2. But here at the Krebs cycle, right, we're making a constant amount of both. And that shouldn't produce a range. The problem is this. Not all NADHs are made in here where electron transport and chemiosmosis are occurring. We have one NADH that's made here. And NADH can't move across the membrane in here. So what has to happen is NADH comes up to the membrane and it interacts with a surface protein that takes its electron and shuttles it across, but just the electron. And when that electron gets shuttled across, it can either be picked up by NAD+ or FAD+. If it's picked up by NAD+, that's awesome, and we get 36 ATPs. If it's picked up by FAD+, it's not as awesome, and we only get 34 ATPs. Okay? So the reason that there is a range is because we create an NADH here, the first step in the Krebs cycle. And when that electron gets shuttled inside of the mitochondrion, it can be picked up by either NAD+ or FAD+. That's a really, really good short answer item, by the by. Want me to say it again?
Yeah.
Okay.
Thank you.
There's a range of ATP created at chemiosmosis because we create one electron carrier in between the membranes of the mitochondria, right? We create NADH here. That's the first step in the Krebs cycle. And that electron gets transferred from that NADH across the membrane to another electron carrier inside. And it can be transferred to either NAD plus or FAD plus. If it's transferred to NAD plus, we create 36 ATP. If it's transferred to FAD plus, we create 34 ATP. Yeah, Sophia.
Let me repeat that.
Okay.
So like, so it just travels to right there, but like there's like a, I'm just not getting like how does it go
So
through?
this molecule,
Yeah.
NADH, it can't move across that membrane,
Yeah.
but
so
it needs to get that electron across the membrane.
Okay.
So it transfers its electron to a protein embedded in that membrane,
Okay.
and it just shuttles it across, and something else has to pick it up on the other side.
Okay.
And it can either be picked up by another NAD plus or by an FAD plus.
Which
Okay.
was created in the,
Yeah, which we're creating over here. So there's a whole bunch of NAD plus and FAD plus just floating around in here, waiting to pick up an electron.
Okay.
And whichever of those two happens along to pick it up, we'll grab it. Yeah.
So was the creation of NADH and FADH2, are those two different adaptations that we've made, or they kind of just happen simultaneously?
Certainly they're a product of natural selection. Yeah.
I'm just wondering why there's two different ones that were created.
It's a wonderful question. And it could be an evolutionary constraint. There could be some other adaptive reason for it, but I don't know. Yeah, Adam.
So when it's transported across that membrane through the protein, it's picked up by an NADH or an FADH2?
It's picked up by either an NAD plus or an FAD plus.
Oh.
Right? To become NADH or to become FADH2.
so in order for both NAD plus and FAD plus, they both have to pick up an electron though to complete the cycle.
Right, yeah. In order to drive the electron transport chain in chemiosmosis, NAD plus and FAD plus have to be carrying electrons over to here. Yep. Yeah.
So is it just gonna be like 40 questions and 5 short answers?
I don't know. I mean, it'll be something like that. Yeah. I don't know if there'll be 5 short answers or 1 long. Like if I had you diagram all of photosynthesis, I feel like that's more than a 5-point item. I feel like that's a 10-point item. And so if I was to have you diagram all of photosynthesis, I would give you 2 10-point items and you could choose which one you wanted to answer. But there will be something like 40 multiple choice items.
Like
Or for the online students, 50 multiple choice items.
photosynthesis or like the photosystem or like the whole thing, like how
Light and dark
far-
reactions.
Light
If I
and
was
dark,
if I was
okay.
to ask you to diagram the light and the dark reactions, that would be more than a 5-point item
Okay.
in my estimation.
Okay.
Yeah.
Just to clarify, so those steps, so the ETC and then the chemiosmosis, when the NADH is or the FADH2 is dropping its electron, that's inside the inner membrane?
Yeah, that's in here. I showed it on the wrong side, but yep, it's dropping it off inside the inner membrane, and then those electrons are being used to pump protons. In fact, let me change that. So you can see that a little bit better. Okay, so what content from this so far would you like to see on the exam? Yeah, Linda.
Could On your exam, you know, like at the back,
Uh-huh.
when you want us to diagram these, could you put like, just like a basic outline of like PS1, PS2,
Like give you the structures and then
we
you guys.
can fill in all the blanks?
Yeah, I probably won't do that, I'm sorry.
Okay.
It was a
Yeah,
good
'cause
question,
that's-
but no, I probably wouldn't do that.
Okay.
I'd probably ask you to diagram
make us
and label
draw
the
it.
whole thing.
But
Yeah.
don't ask
So with
questions.
this one, with the cellular respiration, how would you like us to diagram? You wanted to draw mitochondria and show where.
As long as- As long as I know what's going on, I don't really care what the diagram looks like.
Okay.
My guess is it's gonna look something like this, but if your ATP synthase doesn't exactly look like this, it's okay.
So
Okay,
this
that's-
whole thing is cellular respiration.
Glycolysis, Krebs cycle, ETC, and chemiosmosis is cellular respiration.
Oh,
That makes
I love
sense.
that.
Okay, so what's the aerobic part of cellular respiration? It'd be Krebs cycle, electron transport chain, and chemiosmosis. Anaerobic is just glycolysis.
because it needs oxygen. I know it doesn't.
Correct.
That isn't glycolysis, that's chemiosmosis.
The second, the ATP.
Correct, yeah, this right down here, this is chemiosmosis, and this is electron transport chain. I had to erase glycolysis for room. What other content would you guys like to see on the exam?
Just this.
Just this?
So, if we study these diagrams and know the steps, throughout all of these diagrams, we will do well.
Yeah.
That's
that.
That's hard, yeah.
Well, like, for the multiple choice, is it mostly like based off of the diagrams or
I
like?
haven't even started writing the multiple choice,
Okay.
but you guys, when I write my exams, what I have is, on one of my monitors, I have the PowerPoints, and on the other monitor, on another monitor, I have my Word document. And I'm looking at the PowerPoints, and I'm creating questions from the PowerPoints.
Okay.
That's how I write my exams.
A good question might be, what is the purpose of each?
Yeah, I expect a lot of those kind of
What is
questions.
the product?
Or which of the following stages has the purpose of completely digesting sugar to create a whole bunch of electron carriers? And you guys would say, that's Krebs cycle.
Got it.
Yeah? Okay, so no purpose, no product. How many ATPs are created at this stage? What are we creating at this stage? That type of stuff.
Okay. I can do that.
Yep,
Is this step 4 of,
this would be 3, electron
and then
transport,
4?
and this is 4. Right. Okay, I know you guys are chomping at the bit to get out of here, but let me just go back to the PowerPoints one more time.
Wait, can you go over that was one more time?
Just one more time?
Yes,
Sure.
please.
So when we're creating electron carriers, and this could just as easily be FAD+, what we have is an enzyme that binds to both NAD+, that carrier, and then some substrate that's going to be donating an electron. Right, and so what's happened here is NAD+ is bound to the enzyme in one of the active sites. And then that substrate that's carrying that extra electron is bound in the other active site. And then this enzyme is going to manipulate both of those molecules a little bit so that that electron gets transferred over onto NAD+ to become NADH. And once that happens, they're both just released. Now this could be something like pyruvate and pyruvate and NAD+, right? Because we know in that first stage of the Krebs cycle, we take pyruvate and we break off a carbon. And when that happens, it liberates an electron, and that electron gets transferred over to NAD+. This is a very stylized version of the Krebs cycle. Now, after acetyl-CoA is created, right, after we've taken those 2 pyruvates and stuck them onto coenzyme A, coenzyme A brings those 2 carbons inside, very inside of the mitochondria. And those 2 carbons are transferred onto oxaloacetate. And it creates a 6-carbon molecule like this. And then what we're going to do is we're going to take those molecules and start breaking them off, or those carbons and start breaking them off. And so here, we break off that top carbon, create some carbon dioxide and an NADH, so that we end up with a 5-carbon molecule. And we're going to do the same thing right here. We're going to take one of those carbons, break it off, create another carbon dioxide, and then another NADH right here. And this is where that molecular magic happens, where we get GDP that transfers a phosphate over to ADP to make ATP, right? And then in the last stage, we're going to move the carbons and side groups around on this 4-carbon chain to make back our oxaloacetate. In the process of doing that, we create one FADH2 and one NADH. Now, after the Krebs cycle, we've created a bunch of electron carriers. So we're going to bring those electron carriers over here to the electron transport chain and drop them off and start pumping hydrogens to either side, or to the other side of the membrane, to create that proton gradient. And that's the purpose and the product of the electron transport chain. It creates a proton gradient, right? And then we get chemiosmosis. And through chemiosmosis, those protons passively fall. And again, you guys, this is an important distinction. When the protons fall back through ATP synthase, that is a passive process. It's not active, it's passive. They're passively falling back through ATP synthase, spinning it, and we're able to, through oxidative phosphorylation, create 34 to 36 ATPs. Okay, and this is the diagram of the Krebs cycle and the electron transport chain. When we come back on Monday, we're going to pick up right here and talk about how it is that we can use glycolysis in other kinds of ways. Okay? Any questions? Super, have a great weekend, and I'll see you guys on Monday.